Close your eyes. Imagine you are a fish. You are in a river. Cool water is moving past your gills. Sunlight reaching down to the surface. Plants growing below. Life feels balanced. Now, something changes upstream. A pipe opens. Sewage, water that looks slightly cloudy, enters your world. You cannot see the molecules. You cannot read lab reports of the water. You can only feel consequences. Scene 1. BOD, the oxygen thief. Based on our previous discussion, answer the first question. What do you think kills fish in polluted water? Is it smell? Is it color? Is it foam? Is it a toxic chemical? Well, sometimes yes, but often the real killer is oxygen collapse. Now, listen carefully because this is how exam questions are written. We know from our previous discussion that when untreated sewage enters a river, it carries with it organic matter, which is food for bacteria. And when bacteria eat organic matter, what do they consume? Oxygen. Now, zoom into your gills as the fish. You try to pull oxygen from the water, but there isn't any because trillions of bacteria are already using it. You begin to gasp. This demand for oxygen by microorganisms breaking down organic matter is called Biochemical Oxygen Demand, BOD. Let's break down the word. Bio implies life. Chemical implies chemistry or reactions. Oxygen demand means how much oxygen is needed. BOD measures how much oxygen is demanded by microbiological beings when they eat organic material. BOD does not measure oxygen directly. It measures the potential of a food substance or organic waste or pollutant to remove oxygen while getting eaten by microbes. BOD is a future oxygen demand. It is a prediction of oxygen stress. BOD is a property of the waste itself. Just like gasoline has the property of being flammable, organic pollutants have the property of demanding oxygen when microbes eat them. So when a lab reports BOD equals 200 milligrams per liter, it means this wastewater contains pollutants that when biologically degraded, in other words, when eaten by microbes, will require 200 milligrams of dissolved oxygen from the water. BOD is indirectly used to measure the strength of the organic pollutants. Instead of directly weighing or measuring the amount of organic pollutants in wastewater, we measure how much oxygen will be consumed when we try to get rid of the organic waste by allowing microbes to eat them. BOD is a characteristic of the wastewater itself. It is telling you how hungry this wastewater is for oxygen. Now, imagine you are the fish again. You try to breathe, but trillions of bacteria are digesting this organic matter. The more amount of the organic waste, higher is its BOD, and the more oxygen disappears. This is why BOD is sometimes called oxygen-demanding waste. Now, here is the exam-level clarity. DO, or dissolved oxygen, is equal to how much oxygen is currently in the water. BOD is how much oxygen will be consumed if microbes break down the waste. DO is the present condition of oxygen in the wastewater. BOD is the extra oxygen demand potential of the wastewater. Exams test this difference constantly. If a question says, which parameter measures the oxygen required by microorganisms to stabilize organic matter? Answer, BOD. If it says, which parameter indicates biodegradable organic strength? Answer, BOD. If it says, which parameter is associated with oxygen depletion in receiving waters? Answer, BOD. If it says,
Which parameter indicates the amount of biodegradable organic matter in wastewater? Answer: BOD. Notice the pattern: biodegradable, organic, oxygen required, strength of wastewater. These words signal BOD. Now let's connect this to your job. In a wastewater treatment plant, raw influent wastewater may have BOD of 200 to 300 milligrams per liter or higher. After treatment, BOD of the effluent wastewater may be required to be below 25 milligrams per liter, which is the monthly average in many permits. Why that number? Because rivers have limited availability of dissolved oxygen inside them. If you discharge high BOD wastewater, the receiving river can lose oxygen, and the river may not be able to replenish the lost oxygen to save its fish. Now, let's not forget the truth we understood in the previous lecture. The truth that river pollution is not nature's fault. Nature already tries to clean wastewater. When sewage enters a river, bacteria in the river begin to degrade the organic waste in the sewage naturally. The river is therefore a natural biological reactor, but it still fails to handle the sewage wastewater. Despite having microorganisms that can break down the organic waste from the sewage, the river is unable to break down all of this waste while maintaining the delicate lives of the plants and animals that depend on it. On top of that, the river does not naturally store enough dissolved oxygen to allow the complete degradation of organic waste. Because of that, it ultimately leads to the death of microorganisms once all the dissolved oxygen in the river gets depleted. Now, here is the genius of wastewater treatment engineering. We do not just stop biodegradation in the river, instead, we learn from it and we relocate it. Instead of letting microbes consume oxygen inside the river, we bring the wastewater into a controlled tank. We take the reaction away from fish, and we perform it inside a closed box. That box is called the aeration tank. In an aeration tank, we provide massive blowers force air into water, and diffusers that create air bubbles and continuously supply excess dissolved oxygen to the water. Microbes are allowed and encouraged to grow intentionally. They are allowed to feed on all the organic waste and consume all the dissolved oxygen and multiply rapidly to their heart's desire. What was dangerous in the river becomes powerful in the aeration tank. Thus, we are not fighting biology, we are engineering it and letting it happen in a controlled manner. We take the natural biodegradation tendency and supercharge it under safe conditions. And what are we removing in that tank? BOD. We are allowing microbes to consume the oxygen-demanding pollutants inside the plant, so they don't demand oxygen later in the receiving river. That is the primary role of the aeration tank regarding BOD. Remove the oxygen hunger before discharging to the receiving river. If I say, where should biodegradation happen? You say, in the aeration tank. If I say, why not in the river? You say, because the river cannot supply unlimited oxygen. If I say, what is your primary biological responsibility? You say, reduce BOD before discharge to the receiving river. You are now thinking like an operator. Now that we understand how we engineer biology to control oxygen demand, let's move to another invisible enemy, one that doesn't steal oxygen chemically, but one that physically blocks light and clogs life in the river. Let's talk about suspended solids. Scene 2. TSS and turbidity, the light blockers and gill cloggers. Now imagine you are still that fish, but this time oxygen is okay. You can breathe. Instead, the river changes color. Muddy water rushes in. What is making it muddy? Tiny things, not big trash, not obvious objects. Tiny bits of organic matter, tiny food particles, clay fibers, paper fibers, microsolids, silt and grit fragments. They don't dissolve in water like salt. They don't vanish in the water the way sugar does. Instead, they float in the water. They drift inside the water. They don't sink to the bottom. They stay suspended and move wherever the river carries them. These particles are called total suspended solids, TSS. Suspended, not dissolved, because they are floating in the water. Think of a simple daily example. If you take a glass of water and mix in a spoon of soil, the water turns brown. The soil does not disappear. It stays as tiny brown particles floating and drifting. If you leave the glass still for some time, what happens? The soil slowly sinks and settles to the bottom. That is suspension. Suspended solids are particles that are small enough to stay floating for some time, but heavy enough that they can eventually settle down at the bottom. They are not dissolved chemicals. They are physical particles moving with the water. And because they are physical particles, we measure them by weight. That's why TSS is reported in milligrams of solids per liter. That's exam language. If you see mg/L solids, think TSS. Now the bigger question. Why does TSS matter? What does it do to a river? Two major things. First impact on river, sunlight gets blocked. Plants underwater need sunlight for photosynthesis. When TSS is high, the water becomes cloudy. Light cannot travel deep. Plants get shaded. If there is less sunlight, plants are not able to produce enough oxygen because their photosynthesis slows down. When less oxygen is produced during the day, the ecosystem weakens because there is less oxygen available for aquatic animals to breathe. That is how high TSS can weaken the ecosystem, because it turns the river into a darker, poorer habitat. This is why operators care about clarity, not because it looks pretty, because sunlight is life support for the river. Second impact, gill clogging. Now zoom back to you as the fish. Water flows through delicate gill filaments. These gills are not armored. They are soft. They are thin. Now imagine the water is full of tiny abrasive particles. Those particles rub. They clog. They irritate. It is like breathing through a very dusty air mask. That is high TSS. So while BOD kills by stealing oxygen chemically, TSS harms by attacking physically. It is a different type of enemy. Now, another word you will see on exams, turbidity. TSS is the weight of solids present in wastewater. Turbidity measures how cloudy is the water, not weight. It measures how much light is scattered by particles. It is often reported in NTU, nephelometric turbidity units. So think like this. TSS equals how much solid mass is present. Turbidity equals how cloudy it appears. On exams, if you see the words light penetration, cloudiness, or NTU, think turbidity. If you see milligrams per liter of solids, think TSS. If you see suspended solids, solids concentration, think TSS. Operator role, where TSS becomes a job skill. Now connect this to the plant. In treatment plants, solids are supposed to be captured and removed because high TSS in effluent causes cloudy river, blocked sunlight, habitat stress, gill irritation, and regulatory violation. This is why permits often have TSS limits for effluent wastewater, because solids don't just look bad, they physically damage ecosystems. So if I say light blocker and gill clogger, you say TSS. If I say milligram per liter of solids, you say TSS. If I say NTU and cloudiness, you say turbidity. You just learned parameter recognition the way exam writers expect. And now we shift to the next invisible enemy. BOD steals oxygen, TSS blocks light and clogs gills. But the next one? It can look like nothing is wrong. Clear water, okay oxygen, and still your body feels poisoned. Let's talk about ammonia.
Scene 3. Ammonia, the silent chemical stressor. Stay in the river. You are still that fish. You survived oxygen collapse. You survived muddy water. Now, the water looks clear. Light is reaching the bottom. Oxygen feels fine. You take a breath. And yet, something feels wrong. Your body feels stressed. Your gills burn. Your movements slow. You are not suffocating. You are being poisoned. But by what? Step 1. The source of ammonia. Let's zoom upstream. Where does ammonia come from? Urine, protein breakdown, human waste, animal waste. When organic nitrogen in waste breaks down, one of the products formed is ammonia, which is NH3 or NH4+. Ammonia contains nitrogen, and nitrogen is not always harmless. Now here is something important. Ammonia in water can exist in two forms, ammonium or NH4+, which is less toxic or safer, and free ammonia or NH3, which is very toxic to fish. We will study the chemistry later when we discuss pH and temperature. For now, understand the effect. Free ammonia interferes with fish respiration. It disrupts oxygen transfer at the gills. It damages tissue. It stresses metabolism, causes death of fish. So even if dissolved oxygen in the river is normal, high ammonia can still kill fish. This is a different kind of enemy. BOD steals oxygen. TSS blocks light. Ammonia attacks the fish's gills. Step 2. Ammonia's second hidden effect. But ammonia has a second danger. Remember what we learned earlier. Microbes consume oxygen when they break down pollutants. Well, when bacteria convert ammonia into nitrate, a process you will later learn is called nitrification, they consume oxygen. So ammonia can harm in two ways. 1. Direct toxicity to fish. 2. Creating indirect oxygen demand while being eaten by specific microbes that consider it food. This is why exam questions sometimes phrase it like this. Which nitrogen compound is toxic to fish at elevated concentrations? Answer is ammonia. Or, which parameter contributes to nitrification oxygen demand? Answer is ammonia. Notice how exam language works. Toxicity, nitrogen compound, oxygen demand during nitrification, aquatic life protection. Those phrases signal ammonia. Now, think like we did with BOD. BOD is a property of organic pollution. Ammonia is a property of nitrogenous waste, meaning wastes that contain nitrogen. It tells you how much nitrogen is present in waste in the form of ammonia. It tells you the potential for toxic stress, oxygen consumption during microbial consumption. So when a lab reports ammonia N equals 20 milligrams per liter, it means this wastewater contains 20 milligrams per liter of nitrogen in ammonia form. That is chemical strength. That is ecological risk. That is regulatory concern. Now, zoom out to the plant. In raw influent wastewater, ammonia may be present at significant concentrations, but ammonia are not eaten by the same microbes that attack organic matter or high BOD waste. After proper biological treatment of high BOD waste in aeration tank, ammonia levels are reduced separately because only specialized bacteria eat ammonia by converting ammonia to nitrate. You will study that microbial ecology later. For now, understand the operator's responsibility. If ammonia leaves the plant untreated, fish toxicity risk,
Oxygen demand in receiving water, permit violation. This is why ammonia limits exist in many permits, not because of smell, because of aquatic life protection. If I say fish toxin nitrogen compound, you say ammonia. If I say nitrification oxygen demand, you say ammonia. If I say reduced nitrogen in wastewater, you say ammonia. That is how exam writers think. That is how operators think. Now we have three invisible enemies. BOD steals oxygen, TSS blocks light, ammonia poisons and consumes oxygen during conversion. But there is one more threat, one that does not always kill fish first, one that attacks humans, one that spreads through swimming, irrigation, and drinking water. Let's talk about pathogens.
Scene 4. Pathogens, the invisible travelers. Now zoom out from fish. Imagine a summer day. A child is swimming downstream. A town draws river water for drinking water treatment. The water looks clear, and yet something invisible is moving through the water like a stowaway. Not a chemical, not a particle you can see. Living organisms, microscopic, silent, disease-causing pathogens. They are biological threats. Unlike BOD or TSS, pathogens do not kill fish by oxygen removal. They infect humans. Common indicators used in permits: E. coli, fecal coliform. Pathogens can include bacteria, viruses, protozoa, parasites. Now imagine sewage enters a river. Inside it are microscopic travelers. They don't settle like heavy grit. They don't float like grease. They move with the current. They can attach to particles. They can ride with water. They can survive long enough to reach swimmers, shellfish beds, irrigation intakes, drinking water sources, and beaches. That's why pathogens are not just a plant problem. They are a public health problem. Pathogens in wastewater are living disease-causing microorganisms that come from fecal waste. They are indicators of how much fecal pollutants are in the wastewater. Now listen closely. This is where exams love to test you. Permits often do not require you to test every single pathogen. Instead, they use indicator organisms. Think of indicators like a smoke alarm. Smoke isn't the fire itself, but it tells you fire might be present. Indicators are used because they are easier to test and they signal fecal contamination. Common indicators you will see in permits and exams: E. coli, fecal coliform. So here are some exam recognition cues. If the question says, which parameter indicates potential fecal contamination, answer is E. coli or fecal coliform. If it says, which parameter is used as an indicator of pathogen risk, answer is E. coli or fecal coliform. If it says, which parameter relates to public health and disease transmission, answer is pathogens, whose indicators are E. coli or fecal coliform. This is parameter recognition. This is how exam writers think. Now connect to the plant. BOD is removed biologically. TSS is captured and settled. Ammonia is converted biologically using special microbes. We disinfect pathogens. In the river, these regulatory limits on our enemies protect oxygen, water clarity, habitat, and public health. On the job, operators monitor these numbers daily using lab sheets, SCADA trends, permit reports. On the exam, you must recognize which parameter links to which impact. That is employable knowledge. Now a deeper question. If a wastewater treatment plant meets the BOD limit of its effluent wastewater, does that always mean the receiving river is safe? If TSS is under limit in the effluent, is performance automatically excellent? Is compliance the same as protection? That is where we go next.